Thank you for joining us for today's presentation in which we will be reviewing the pluripotent stem cell culture systems provided by Thermo Fisher Scientific. This presentation was put together to assist you in identifying the culture system which best fits your workflow needs, as well as to provide you with tips for successful culture in each of the systems. The pluripotent stem cell culture system broadly encompasses your cells, such as your ESCs or iPSCs, the medium and extracellular matrix in which your cells are cultured, as well as the passaging reagents which are used in routine culture and expansion of your PSCs. Additionally, cryopreservation reagents play an important role in ensuring cell line security upon expansion. In this presentation, we will be reviewing the tips and tricks for successful pluripotent stem cell culture in the knockout serum replacement feeder-dependent system, as well as the StemPro HESCS-FM and Essential 8 feeder-free systems. Additional information will also be provided regarding transition of existing pluripotent stem cell lines from feeder-dependent to feeder-free culture systems, as well as cryopreservation reagents to ensure cell line security during expansion of your derived pluripotent stem cells. Here we highlight the progression of pluripotent stem cell culture systems over time. Initial PSC cultures utilize the richness of fetal bovine serum to support the health of pluripotent stem cells cultured on feeders. Yet there was a large amount of inconsistency between FBS lots. In 1997, in a move to eliminate the inconsistency of using fetal bovine serum, knockout serum replacement was launched. Since that time, thousands of papers have been published, and this well-established system is still used today. To further simplify the system and eliminate the need for feeder cells, researchers then moved to mouse embryonic fibroblast or human fibroblast conditioned medium to support culture of their cells. Further refinement provided for defined feeder-free growth medium containing bovine serum albumin, yet there were still lot-to-lot inconsistencies noted with the bovine serum albumin component. Finally, Essential 8 was developed by Gibco Chen and colleagues in James Thompson's lab. This system provides a xeno-free medium and matrix for feeder-free culture of PSCs. Together, the system reduced the variability compared with existing feeder-free culture media and provided a cost-effective and robust system for culture of PSCs. In the following slides, we will summarize the feeder-dependent and feeder-free culture systems provided by Thermo Fisher Scientific, as well as the best practices for culture in each system. As mentioned earlier, knockout serum replacement, or KSR, is used for robust feeder-dependent culture of PSCs. This system is provided as a kit comprised of a basal medium, the knockout serum replacement, as well as a basic FGF to support the maintenance of pluripotency of your stem cells. Additional components which are not included in the kit that are added to the complete medium include MEM non-essential amino acids as well as beta-mercaptoethanol. As this is a feeder-dependent culture system, PSCs are grown in colonies on a layer of irradiated mefs, which are seeded onto attachment factor. In the image on the bottom left of your screen, you can see the iPSCs growing on a layer of mouse embryonic fibroblasts. There are two commonly used methods for passaging your cells, either manual passaging or enzymatic. In manual passaging, typically syringe needles or a StemPro Easy Roller passage tool are used. Alternatively, for enzymatic passaging, collagenase 4 or disbase 2 are used. Examples of these passaging methods will be reviewed on subsequent slides.
Looking at the workflow for KSR-based medium culture, plates are initially coated with attachment factor onto which the mefs are seeded. The pluripotent stem cells are then seeded onto this mef layer in the presence of KSR-based medium. And over time, routine manual cleanup of the cultures is carried out to remove areas of aberrant differentiation. And finally, the cells are passaged. Here we will cover some considerations for the system to help you ensure successful culture of your PSCs. For matrix considerations, the feeder layer may be prepared in advance of culture, providing experimental flexibility, but it should be used within 4 days of preparation. Additionally, to further support the health of your PSCs when using the KSR-based system, it is recommended that you condition your medium for a few hours ahead of splitting your PSCs to ensure maximum post-passage survival. It is important to always remember the stability of the medium system. Once prepared, the KSR-based medium is stable when stored at 4C, protected from light for up to 2 weeks. Basic FGF should only be added to the culture medium immediately prior to addition to your cells at each feeding, and daily feeding is recommended for the system. Once your colonies become dense, large, or overcrowded, such as the representative image shown here, then cells should be passaged. Additionally, cells should be passaged when increased differentiation occurs or when the feeder layer is 2 weeks old to maintain health of your culture.
The following images provide some insight into areas for removal during manual cleanup of colonies. The colony on the left has a heterogeneous core, loss of smooth defined border, as well as significant aberrant differentiation highlighted in the red outline. This colony should be removed manually to maintain health of your PSC culture. The image on the right shows a colony with a homogeneous core with some aberrant differentiation occurring on the top of the colony, which should also be removed during manual cleanup. In addition to the aberrant differentiation which can occur on the sides of the colonies, differentiation can also be observed in the center of colonies and should be removed during manual cleanup. 2 examples are shown here, with the far left image showing flattened cells in the middle of the colony that should be removed. And the figure on the right shows the development of an embroid body in the center of the colony, which should also be removed. Regular cleanup of your KSR-based feeder-dependent cultures is essential to maintain the health and quality of these cultures. In the event that significant aberrant differentiation has occurred throughout your culture and only a small portion of the colonies show normal pluripotent morphology, colonies can be positively selected and transferred to new feeder plates for continued propagation. In the images shown here, we see the progression along this continuum. In the far left, you can see a large amount of aberrant differentiation in the colony circled in red, with a good pluripotent colony shown by the green circle. Upon identifying the undifferentiated colonies, colonies are scored with a syringe needle and subsequently scraped using a pipette tip and transferred to a conditioned feeder plate. If the culture is largely undifferentiated, then areas of differentiation should be removed and cells should be passaged using the methods highlighted on the following slide.
For bulk passaging of undifferentiated pluripotent stem cells, there are manual or enzymatic options available. For manual passaging, we recommend the use of the StemPro Easy Passage disposable stem cell passaging tool. This tool provides a quick and efficient manual passaging method which provides consistency and uniformity of size of colony clusters. Following manual cleanup of differentiated colonies and subsequent washing for the removal, the Easy Passage tool is ran in one direction along the plate in rows, and then turned 90 degrees and ran across the plate again in rows. On the top right is a representative image showing the consistency of fragment sizes produced using this tool. Cells are then gently scraped from the surface of the plate and transferred to a new plate, generally at a 1:3 to 1:6 split ratio. Alternatively, bulk passaging can be performed via the treatment of undifferentiated PSCs with either collagenase 4 for 30 to 60 minutes at 37C or DisPhase 2 treatment for 5 to 15 minutes at 37C. Incubation with these enzymes should be stopped when the colony edges begin to peel from the plate. Cells are then subsequently triturated to break up the colonies into small clusters of 50 to 500 cells for propagation. We will now move on to review the two feeder-free pluripotent stem cell culture systems available from Thermo Fisher Scientific. The first feeder-free PSC culture system that was launched by Thermo Fisher Scientific was the StemPro HESC SFM system. This system is a fully defined, serum-free system which is provided as a medium containing supplement, a bovine serum albumin additive, and a basal medium. Much like the KSR-based medium, additional medium components are required, including 2-mercaptoethanol as well as FGF-Basic. Cells cultured in this medium system are seeded onto Geltrex matrix and routinely passaged using collagenase 4 incubation for 3 to 5 minutes. The image in the lower left shows representative morphology of pluripotent stem cells cultured in the StemPro HESC SFM system, showing a homogeneous core and high nuclear to cytoplasmic ratio of the PSC. Looking at the workflow for StemPro HESC SFM medium cultures, plates are initially coated with Geltrex. PSCs are then seeded onto the Geltrex in the presence of StemPro HESC SFM. Routine manual cleanup of the cultures is carried out with removal of differentiated areas like that discussed for the KSR-based system. And finally, cells are passaged. Here we will cover some important considerations for working with the system to help you ensure successful PSC culture. For matrix considerations of working with Geltrex, the Geltrex solution should be thawed overnight at 4 degrees C. When working with Geltrex, it is imperative to keep it chilled to prevent polymerization. Therefore, Geltrex should be transferred on ice from a 4C refrigerator and diluted into pre-chilled DME-F12. A dilution of 1 to 100 is recommended for PSC cultures. Much like the KSR-based medium, the stability of StemPro HESC SFM, excluding the addition of the FGF basic, is 14 days at 4C. Medium should be discarded past this time. Immediately prior to addition of to the cells, FGF basic should be added to the culture medium. It is recommended to passage the cells when they become overcrowded or too large. Finally, we will cover the properties of the essentialate medium. This system is further streamlined to ensure consistent performance and is a xeno-free, feeder-free culture medium system. Essentialate medium comes as a complete formulation comprised of a basal medium and supplement and does not require the addition of other media components. There are multiple offerings for matrices that can be coupled with the essentialate medium system depending on your application and system needs. There are 2 xeno-free matrices that we offer, including a fragment of recombinant human citronectin and full-length recombinant human laminin 521, which are both recombinantly expressed. If a xeno-free system is not required, the cells can be cultured on Geltrex substrate as well. Depending on your downstream application of interest, 2 passaging reagents are commonly used with the system. Bursine, an EDTA solution, is used for routine culture of pluripotent stem cells. For applications in which cells need to be singularized, such as in high-throughput screening or differentiation protocols, cells can be passaged using EEE Select. On the next slide, we will review the properties of the various matrices to assist you in your selection. Looking at the workflow for essentialate medium cultures, plates are initially coated with extracellular matrix of interest. The pluripotent stem cells are then seeded onto the ECM in the presence of essentialate medium, and cultures are maintained by daily feeding. The cultures do not require manual cleanup of differentiated areas prior to passaging. The table provided below will assist you in identifying the appropriate matrix to fulfill your workflow needs. As mentioned earlier, if a defined surface is required, then recombinant human vitronectin or recombinant human laminin 521 should be used. Always keep in mind the thaw temperature, stability, and diluent for each of the systems as well to ensure matrix integrity. Initial coating concentrations are provided but may need to be optimized depending on your cell line or downstream application. And of the matrices offered, recombinant human laminin 521 is the only matrix that allows for robust single-cell passaging in the absence of ROCK inhibitors. Recombinant human vitronectin and Geltrex require the addition of ROCK inhibitors or a pro-survival small molecule containing cocktail RevitaCell to ensure optimum cell survival upon single-cell passaging. We will now move on to medium considerations for the Essential 8 system. The Essential 8 supplement, which is provided as a 50x supplement, should be thawed at room temperature for approximately 1 hour. Alternatively, you may thaw the supplement overnight at 4 degrees C, but you should not thaw it in a 37-degree water bath. Upon addition of the supplement to the basal medium, the complete medium is stable for 14 days when stored at 4C protected from light. Once reconstituted, the freezing of the complete medium is not recommended due to the sensitivity of this formulation to multiple freeze-thaw cycles due to the growth factors. Prior to feeding of the pluripotent stem cells, media should be allowed to come to room temperature gradually, whereas pre-warming of the medium in a 37 degrees C water bath is not recommended. For the Essential 8 medium system, daily feeding is required. However, recently we launched the Essential 8 Flex Medium Kit, which provides a weekend-free feeding option for our xeno-free systems.
Here we will cover passaging considerations for the Essential 8 medium system. Pluripotent stem cells should be passaged upon attaining an approximately 80 to 85% confluent culture, and cells should not be allowed to become overly confluent, as this will impact their post-passage cell survival. Routinely, cells are passaged using clump passaging methods using Versine or 500 micromolar EDTA for this system. You should limit exposure to the Versine solution, as this incubation is cell line dependent, varying from 3 to 5 minutes when incubated at 37°C or 5 to 7 minutes when incubated at room temperature. Cells should only be allowed to form small holes within the colonies, as shown in the image on the bottom. You should also avoid over-trituration of your pluripotent stem cells following Versine passaging to prevent them from breaking into small single cells, which would not recover in the absence of ROCK inhibitor. If single cell passaging is required for your downstream applications such as differentiation or high throughput screening, then pluripotent stem cells can be passaged using EEE Select. As discussed earlier, recombinant human laminin 521 is the only matrix which efficiently supports robust single-cell passaging of PSCs in the absence of ROCK inhibitor. If passaging using single-cell passaging techniques onto Geltrex or recombinant human vitronectin surfaces, then a ROCK inhibitor or RevitaCell supplement should be added at the time of seeding. Here we will review the properties of the Gibco RevitaCell supplement. This supplement was designed to assist in post-thaw recovery of PSCs, as well as to enhance single-cell passaging recovery in Essential 8 medium. The RevitaCell supplement is provided as a 100x bullet, which, when added to the medium for the first 24 hours post-thaw or post-passage, significantly enhances cell survival. It is an animal origin-free and chemically defined formulation, which is perfect for use with Essential 8 medium. This solution is comprised of a ROCK inhibitor coupled with antioxidants and free radical scavengers in order to support your PSCs during stressful transitions. Here we show some data to support the use of RevitaCell supplement in single-cell passaging of PSCs in Essential 8 medium when cultured on recombinant human vitronectin surfaces. On the left uppermost panel, you then see the IncuSite Zoom monitored confluency over time post-passage. The Essential 8 alone condition does not recover well, whereas the Essential 8 with RevitaCell supplement, when RevitaCell supplement is only included for the first 24 hours, recovers very robustly. Single-cell passaging using RevitaCell can be used for multiple passages. Here we show that normal morphology, pluripotency as assessed by OCT4 and TRA1 to 60 staining, as well as normal karyotype are maintained for up to 30 single-cell passages using RevitaCell. Now we will take a look at the morphological changes which occur on Essential 8 medium using different passaging methods and substrates. Here we present EDTA passaging of PSCs in Essential 8 medium on recombinant human vitronectin substrate. On day 0, we can see small clusters above the culture surface, and on day 1, those small clusters have settled down to form small colonies on the plate surface. You will begin to see them grow together as we proceed forth to day 4. Here we show single-cell passaging of pluripotent stem cells in Essential 8 medium on recombinant human vitronectin. In this specific instance, we include RevitaCell supplement for just the first 24 hours post-passage. So on day 0, you see largely single cells in the field of view, and then on day 1, you can see small stretched cells that have settled down. And on day 2, you still see some spiked morphology, which resolves upon reaching confluency. It's important to note that the spikedness of colonies may be exaggerated post-thaw using RevitaCell supplement. Much like in culture on recombinant human vitronectin, with RevitaCell supplement, recombinant human laminin 521 performing single-cell passaging in Essential 8 medium system shows very similar morphology, with stretching of cells toward each other forming spiked edges on the colonies. This again resolves itself as the cells reach confluency. Here we provide a summary table highlighting the different properties of the various media systems covered within this presentation. This table will assist you in identifying the correct system for your workflow needs. As discussed, if you need a xeno-free solution, then Essential 8 medium or the Essential 8 Flex, which has a weekend-free option, is the right choice for you. The knockout serum replacement medium kit, which uses feeders, is the most robust system, but also has the highest complexity. Take a moment to review the table to decide the best system for you. Now we will review some tips for successful transitioning of existing feeder-dependent cell lines to feeder-free culture systems using the Essential 8 Adaptation Kit. The Essential 8 Adaptation Kit contains Essential 8 medium as well as a bullet of recombinant human laminin 521. In adaptation of proliferating feeder-dependent cultures, the cells are enzymatically passaged, they're harvested and washed, there's an additional wash step, and finally, cells are seeded into the feeder-free system on the matrix of interest. Here we show OCT4 whole well imaging staining 5 days post that transition into Essential 8 medium on various substrates. What you quickly observe is that the laminin 521 condition has optimum post-passage cell survival. This is further highlighted by the plot shown here, with the incucyte zoom percent confluency over time indicated. What you can see is that the laminin 521 shows the most robust performance in transitioning of cells from a feeder-dependent to feeder-free context. Finally, we'll provide you some information on the Gibco PSC Cryopreservation Kit in order to enhance your cell line security. The PSC Cryopreservation Kit comes with two components. It has a PSC cryo medium for cryopreservation of your cells and is also sold with a RevitaCell supplement for recovery of your cells to optimize the post-thaw recovery. The PSC cryo medium is a ready-to-use formulation which is xeno-free, whereas the RevitaCell supplement that was covered previously, again, is an animal origin-free solution and is chemically defined. Here we show some data of cryopreservation of KSR-based cultures in the PSC Cryopreservation Kit, as well as using a homebrew method of using medium A plus medium B. What you can see is that in KSR medium alone recovery instances, you see nice recovery of the cells 6 days post-thaw. However, when RevitaCell is included for the first 24 hours post-thaw, you see a significant boost in recovery for both cryopreserved samples. Here we see the cryopreservation of feeder-free cultures using the PSC Cryopreservation Kit. What we can see on the leftmost panel is that we're looking at the 24-hour post-thaw viability for each of the solutions. The serum-containing solution using FBS with 10% DMSO is shown first, whereas serum-free Thermo Fisher Scientific solutions are shown next, followed by serum-free competitive solutions. What you can see is that the PSC Cryopreservation Kit, which is shown in the blue circle, provides optimum post-thaw viability. This allows you to have quicker time to experiment, as indicated by the plot shown below, with the percent confluency reaching passaging confluency much quicker in the PSC cryopreservation instance versus a best-in-class coupled with the 10 micromolar Y27632. The PSC cryopreservation kit is also very versatile for cryopreservation of your PSCs, allowing you to cryopreserve them either as single cells or using clump passaging methods. Here we present a table comparing the PSC cryopreservation systems. As mentioned in this presentation, the PSC cryopreservation kit comes as a ready-to-use formulation that contains a recovery component, is xeno-free, is manufactured also in a cGMP compliant facility. Currently, we do not have a cell therapy systems version available, but the performance of this system is superior. For SyntheFreeze is an alternative cryopreservation medium, which you can also use. It comes as a ready-to-use formulation. It is not supplied with a recovery component, but can be used in conjunction with RevitaCell, which is sold as a standalone. It is a xeno-free formulation, is not manufactured in a cGMP compliant facility, but we do have a cell therapy systems available for this cryo medium. With the information that's been provided to you in this deck, we've covered the pluripotent stem cell media systems available through Thermo Fisher Scientific. Using these systems, you are able to maintain your cells in routine culture and maintain normal PSC properties, including morphology and pluripotency with SSEA4, OCT4 expression, normal karyotype, as well as maintain the tri-lineage differentiation potentials of your pluripotent stem cells for your downstream differentiation experiments. If you are looking for additional opportunities through Gibco, please join us for our hands-on training courses offered at one of our 3 sites in either Carlsbad, California, Glasgow, Scotland, or Frederick, Maryland throughout next year. This course offers a 3-day hands-on training course, which is covering lecture as well as hands-on instructional information. It's limited to a maximum of 8 attendees per course for very personalized attention. Alternatively, you can join us at one of our virtual training labs which cover pluripotent stem cell culture. You can learn it in a virtual environment and attend from anywhere. We have opportunities coming up at the end of this year and early next year. For more information, visit us at www.thermofisher.com/PSC workshop. Thank you for joining us for this session of 24 Hours of Stem Cells.